We're going to start talking about the cellular innate immune response today, which is covered in Chapter 4 of your textbook. Just to let you know, that section of Chapter 4 that I listed pages-wise on the syllabus is really for today and for Wednesday. So I'm going to be hitting parts of it today and parts of it on Wednesday. So you will hear information about that stuff on Wednesday if you have already read through some of it. Last time we talked about proteins in the innate immune system. And we saw these proteins that are able to act immediately within the first 4 hours following infection. They are preformed proteins, so they're able to react immediately. They don't need to do anything in order to start their reaction, and can potentially eliminate pathogens quite quickly. However, sometimes those antimicrobial peptides or complement are not enough. And so sometimes, in fact, the pathogen is not eliminated, and we need an additional phase to the response. And so for the next few days, we're going to be talking about this later innate immune response that's happening usually within the first 96 hours or so. That is going to be done more by cells of the innate immune system as opposed to preformed proteins. And these cells, of course, as you might guess, because they are shown here to start responding at about 4 hours after infection, may need to do a little bit of something to get ready before responding. And so we're going to talk through this whole process of how these cells respond and then how they get rid of the pathogen today. So that's going to be our plan for this discussion. This process that we are talking about also has another name. And the name of this process that we're going to talk about is inflammation. Inflammation is a word that a lot of people have heard. It's used in a lot of settings, and people often think about inflammation or this inflammatory response as being a negative, though as I'll show you a little bit on the next slide, it's not always a negative. It also has a lot of positive aspects. But inflammation was first in our kind of written descriptions of immunology described by the Romans. So this is one of those things, like I told you about at the beginning, where people have been observing their bodies for a long period of time and have been able to see what's going on and describe some immune reactions for a long period of time. So if you think about inflammation, I'm sure you have all experienced inflammation before, there are some signs that you frequently see. So what are those major signs of inflammation? Yep, Alina. Redness. Which the Romans referred to as rubor. All right, what else? Let's see, Akuya. Swelling. And they referred to that one as tumor. Let's see, Emilio. Heat. Heat or calor. And there's one more. Yep, Calvin. Pain. Dolor, that's supposed to be a D. And so these are sort of the 4 key signs of inflammation. They're very easily observable, and as we talk through the cells and molecules that are going to be part of this process, we are going to see how these 4, what are known as cardinal signs of inflammation, actually relate back to the events we're seeing in this response. So as I said, if you watch TV or listen to different people talking about inflammation, it's, the big bad thing that is causing all of your problems. that you definitely need to get rid of. You always think of it as being a big problem. But inflammation is an important physiologic response. Inflammation can happen as a result of infection, as a result of injuries like wounds, or as a result of tissue stress. And if things are going well, this inflammatory response is going to give you defense against your infection. It's going to help with tissue repair, and it's going to help you return to homeostasis. So all of this is actually a really, really good thing. So we're going to walk through these steps of this inflammatory response. One thing I will point out is that the classic response I'm going to talk about today is most frequently thought about as a response to invading bacterial pathogens. It doesn't have to be bacteria. It can be fungal. It can be viral. We can have lots of arguments about that. We can read Tyler's thesis if we want. And learn a lot about some of these distinctions. There is sort of a classic viral response, which I'm going to talk more about on Wednesday. So today is sort of a classic bacterial response, although yes, it can happen in response to many types of pathogens. And it can happen in following, say, tissue damage, as you see here, that might allow bacteria in. And so you can imagine a wound. I think that every immunology exam I took in undergrad and grad school involved a problem where the professor lectured very fervently and hit their hand on the podium and got a wound on their hand and bacteria in it. For some reason, everybody makes their question that way. Don't know why. So you can imagine a break in the skin, perhaps by this weird glass-looking thing, letting in bacteria. The first step that's listed here is that resident sentinel cells are going to release some molecules. I'm going to give you a little bit more of a definition of those resident sentinel cells. Because in fact, those resident sentinel cells are cells you already know about. They're one of the types of cells we learned last week. Those resident sentinel cells are largely the tissue macrophages that we talked about last time. And so by and large, this first step is happening as a result of some macrophage or monocyte in the tissue. One thing that you are going to notice today is I'm going to spend some time talking a little bit about macrophages, and I'm going to spend a little bit of time talking about neutrophils. Students frequently confuse macrophages and neutrophils and some of their functions, and so pay attention as we go through today about what some of the key similarities and differences are in terms of macrophages and neutrophils. So we've got these resident macrophages in tissue, and as our image shows, those resident macrophages are going to release, and again, there's a list of substances here. If we really want to think about that process and what's going on, the first step that we might wonder about is, okay, great, how does the macrophage know that there is a microbe to which it should respond to? How does the macrophage know that there's a bacteria present? If it's going to actually do something, as is shown in box 1 here, it clearly has to know there are bacteria present. Macrophages don't have eyes or brains. Remember last week we were talking about some definitions of how the immune system worked, and you talked about a perceived problem. Perceived does have an awful lot of brain going on to make perception happen. So how does the macrophage know there's a problem that it needs to respond to? This is part of the key to the innate immune response. One of the things that's great about the innate immune response is that it is very, very good at a process known as self-non-self recognition. So our innate immune cells have an ability to recognize biochemical components that are found on foreign organisms that are non-self and potentially respond. So this is something that the innate immune system is very good at. And this is the type of response that is happening here. Specifically, our macrophage is going to use a type of receptor. That receptor is known as a pattern recognition receptor, excuse me, or PRR. The idea is that this is going to be a receptor that can bind to some component of a microbe. We can think of it as a microbial pattern that is present on non-self organisms, but not on self. Typically, as you can see on the right hand of this slide, individual cells have a huge diversity of PRRs. Different cells may have different PRRs. All of the ones that are shown here are shown on the cell membrane, but there are PRRs throughout the cell, in vesicles, in the cytoplasm, perhaps even in the nucleus. So that's not shown here, but you can see all of the cell being defended with PRRs. These are, it says here, encoded in the germline, which will probably not make any sense to you right now, but the basic idea is they're encoded like normal proteins are. And we're going to see some things that are not encoded that way later. You can see some examples here. And the other thing that's really key is that these receptors bind to targets that are not specific to individual microbes. Instead, they are specific to broad classes of microbes. So a pattern recognition receptor is not going to distinguish this year's flu versus next year's flu. That's an adaptive immunity thing, not an innate immunity thing. However, we may see recognition of all RNA viruses like influenza by seeing recognition of the specific type of RNA that those viruses use. So many different microbes could have similar patterns, all hitting the same pattern recognition receptor. The part of the microbe that is recognized also has a specific name, although its name has changed a little bit over time. So some people refer to this target recognized by our pattern recognition receptor as a PAMP. The P at the end stands for pattern, so that's good. And the other letters are pathogen, associated, and molecular. So these are unique parts of microbial pathogens that can be recognized by these receptors. Some examples include things like peptidoglycan, part of the bacterial cell wall. Things like some types of single-stranded RNA, things like flagellin, part of the prokaryotic flagella, things like zymosan, which is part of a fungal cell wall, things like hemozoin, which is an important component of the malaria parasite. So we have many different types of molecules. By and large, these are molecules that are found on broad groups of microbes instead of one individual microbe. Some people have said that the term pathogen-associated molecular pattern is an inappropriate term because pathogen is a type of microbe that causes disease and damage, but there are many microbes that don't cause any problems that still have these things. There are many microbes that are living happily in your body right now in your GI tract that all have peptidoglycan or that have LPS. And so some people say, no, we shouldn't call them PAMPs. We should call them MAMPs, or microbe-associated molecular patterns. And I anticipate at some point we will have a little bit of discussion based on some questions that you all ask about the fact that there are another group of proposed similar molecules known as DAMPs, which stands for damage-associated molecular pattern, where perhaps a cell that is injured may also produce some molecule that can lead to an inflammatory response. The biology of DAMPs is a little more complicated. There's a lot that can be said there, but there's also a lot that's controversial. But just realize that if you sprain your ankle, there is no microbe, but somehow there is still inflammation. It's usually known as a process called sterile inflammation. And so we can think about where exactly the stimulus for the macrophages coming in. Once our PRR recognizes its pattern, whether it's a PAMP or a MAMP or a DAMP, a few different events happen in the cell. What you will notice here and on the next few slides is that I'm being very general about what the PRR is and the signaling that happens downstream of it. That's something I'm going to talk about in more detail on Wednesday. It is also covered in Chapter 4. Here we're going to get big picture. We're going to zoom into some of those details on Wednesday. So one thing that can happen is that when our pattern recognition receptor binds to a microbial pattern, we can have phagocytosis of that microbe occur. So we can have these phagocytic receptors acting. Your textbook talks a little bit about opsonizing receptors here. We talked about the term opsonizing on Friday of last week. In fact, your textbook says, from the Greek, to make tasty, aka butter. And so the first thing that can happen as a result of pattern recognition receptor activity, if that receptor is on the surface of a cell, is phagocytosis. However, this is not the only thing that can happen as a result of pattern recognition receptor activity. One thing I should mention, I forgot to mention earlier, is that there are plenty of other types of cells besides macrophages that do have pattern recognition receptors. They are the most well-studied in macrophages. And the process that we're talking about today involves macrophages and their PRRs. There are plenty of other cells that have them as well. So we can have phagocytosis of our microbe, leading to its degradation. We're going to talk about that a little bit. But in the case of our macrophage, we can also see signaling that leads to changes in transcription. And that will allow our cell to make some new products that can affect the immune system and actually get rid of the pathogen. These products can have a lot of different roles. One of them may have to do with killing microbes. Some of them are going to have to do with inflammation. Others are going to have to do with tissue remodeling and wound healing. Immunologists often ignore that wound healing and tissue remodeling step, and more we are finding interesting ways that it's linked with some of our other more famous types of the innate immune system. And so that's a pretty good current area of research. So at this point, our macrophage, our resident sentinel cell, has now recognized the bacteria and made some changes in transcription and can now actually elaborate products or make products that can start to help us get rid of these microbes. And so you can see now we are ready to release chemoattractants and vasoactive factors. Specifically, these factors are known as cytokines and chemokines. Cytokine is a fancy immunologist term for a secreted protein made by one immune cell in order to communicate with another cell. It's just a secreted protein for cell-cell communication. Many of them, though not all of them, because things can never be easy, are referred to as interleukins between leukocytes, between white blood cells. The point is that these are secreted proteins that are communicating within the immune system. In today's case, we are talking about a subset of cytokines that are known as pro-inflammatory cytokines. They lead to inflammation. I point that out because some students get really confused and decide that all cytokines are pro-inflammatory cytokines, which is not the case. We're going to see many that do other things throughout the semester. But today we are going to see some pro-inflammatory cytokines. Some of the pro-inflammatory cytokines that we are going to see at this point are called IL-1 beta, TNF alpha, and IL-6. We can also see some other products being made by our macrophage. Two of them are enzymes that are not listed here, but that I want to mention because they're going to come up later. So these are enzymes. They're inside the macrophage. They're not being secreted. One of them is called INOS, and we'll come back to INOS. The other is called COX-2. COX-2 stands for cyclooxygenase 2, and it helps the macrophage make lipids that are going to be released. We can kind of we'll call them right now inflammatory lipids. So these enzymes are made by our macrophage, and one other group of proteins are made by our macrophage. These are known as chemokines, which is listed at the top. Chemokine is actually kind of a shortened version of their definition of these proteins. Chemokines are cytokines that are involved in cell movement. They tell cells to move, or they are involved in chemotaxis. And so the official term is chemotactic cytokine. But people have taken chemotactic cytokine, which is really long, and smushed it together to make chemokine. So we also have chemokines that are made. One of them is shown here. It has a few different names. It's not super important. Yes, it is IL-8. We'll see IL-8 later. That's going to be made by this macrophage. These proteins have a number of different types of effects. The biggest effect of our IL-1 beta, TNF alpha, and IL-6 has to do with changes to blood vessels, inducing blood vessels to be permeable and changing that blood vessel diameter. You can see that happening here. So once our inflammatory cytokines, IL-1 beta, TNF alpha, and IL-6 are produced, our blood vessel is going to change dramatically in diameter, which you will notice here, and become much more permeable. So you'll see that the nice junctions that we previously had are now leaky. So we've got changes in vascular permeability and vasodilation. You can see that there are also cells that are now leaving the vessel and going to this area. That's because of those chemokines, those molecules that are telling cells to move, that are involved in chemotaxis. If you were to think through this process, could you imagine any way that this might lead to swelling? Yeah, Molly. More stuff, more cells, more liquid, because all of the liquid of the blood is leaking into this area. So we're going to get that swelling that we see as a cardinal sign of inflammation because of this response. What about the redness? Yeah.
So there's blood, so blood is red.
So we're getting blood flow to that area. What about heat?
It's also blood.
The blood's warm. So in fact, all of the effects that we think of as the cardinal signs of inflammation are really the result of these inflammatory cytokines acting on the local vasculature. The pain is a little more complex, but we do know that many nerves have cytokine receptors. And we can imagine that there is this important evolutionary advantage where if you have inflammation on your hand from getting a cut on your hand from pounding it on a podium, then it's probably a good thing that you know and that you protect it instead of like dragging it around the dirty ground and getting more bacteria. And so in fact, having some type of pain response so that you protect that wounded area makes some evolutionary sense. You can do a PubMed search for someone named Isaac Chiu, C-H-I-U, if you are interested in understanding this response. He was actually one of my classmates in grad school and now does some of the really great work on both pain and itch in response to these cytokines. So we see all of these effects of these inflammatory cytokines. Yeah, Alina?
Is anything in there like more than others? Is there like more something?
Do you mean in terms of the cytokines, or do you mean in terms of like what do you mean?
Everything when it's becoming permeable, is anything coming in more than others?
So there are a lot of fluids and proteins coming in, but there are some unique cells. So I haven't totally focused on the cells that's coming. It is one type of cell that is most frequently coming, though it can be a lot, but there's one type that's really important here. Just so you are aware, last time when we talked about complement, we saw two proteins called C3a and C5a. C3a and C5a act identically to the cytokine, or sorry, to the chemokine that I just told you about. So C3a and C5a basically act as inflammatory chemokines and induce the same kind of permeability, the same kind of migration, all the same effects that we saw with those inflammatory cytokines and chemokines. They signal through receptors that are quite similar to chemokine receptors and lead to a lot of the same effects that we're seeing. So as much as I can try to separate out all of these responses, you can see that they are all very closely tied together. The chemokines in particular are going to be really important for the recruitment of other cell types. The other cell types typically are going to be neutrophils here. And so this is where we're going to see some similarities and differences between macrophages and neutrophils. There is a very specific 3-step cascade that all immune cells use in order to move to parts of the body and leave the blood. The exact proteins vary a little bit, but the steps are the same anytime an immune cell wants to leave the blood. I am going to focus on those 3 steps when we talk about T cells and how they interact in the lymph node, because that's where I usually talk about them. But just so you know, there is a very regulated process that these neutrophils and other cell types are going to go through. And it's the same process we're going to see later to allow these cells to be recruited. So in the end, we see this process of local inflammation. Our person here had an injury to their hand's red. We had changes in vascular permeability, changes in vascular diameter in their hand. We had recruitment of cells in their hand. We got rid of the microbe, and then they lived happily ever after, all better, with no longer a red hand. And those are the good local effects of inflammatory cytokines. Inflammatory cytokines don't just act at the site of inflammation, however. They also act in some other parts of your body. So here is sort of the action that's happening just in the hand with the inflammation. But those inflammatory cytokines can get in the bloodstream and start to act on other organs as well. One of the big effects of those cytokines is an effect that IL-6 has on the liver. IL-6 can act on the liver and induce the liver to make a new set of proteins. They're known as the acute phase proteins. This is called the acute phase response. Some of the proteins that are made in the acute phase response include a protein known as mannose-binding lectin, another protein called C3, another protein called C4, one called C9, one called factor B. Have you guys heard of any of those before? No?
No, never.
Never? What are those proteins? Complement. Those are the complement proteins we talked about on Friday. And so in fact, again, all of these responses are interconnected. So the inflammatory cytokines are going to induce higher levels of complement proteins so that we may be able to further clear our microbe using the complement pathway that we talked about on Friday. C-reactive protein also can have some effects in complement. And in fact, there are a number of different wound healing proteins that can be made here as well. So these inflammatory cytokines can act in the liver and get the liver to make some new proteins that can be useful. These inflammatory cytokines can also act in the bone marrow. I mentioned when we talked about cells of the immune system that you have a huge store of neutrophils in your bone marrow. When inflammation occurs, inflammatory cytokines can act on the bone marrow to lead to release of more of those neutrophils. So again, we have more cells present in order to perform phagocytosis. So we're going to sort of call in the cavalry, we can imagine. We can also see a number of other effects. So you can see the liver effect here, the bone marrow effect here. But there are also changes to things like body temperature and metabolism. The metabolism effects largely support that body temperature change. These temperature changes can do things like make your body less hospitable for bacterial replication. If the temperature was changed of your body long term, there would also be less hospitable replication. But if we do this for a short period of time, this can knock down bacterial numbers. Remember that we talked about this huge problem of how fast microbes replicate and how quickly they can out-replicate us and out-replicate our cells. Any of these processes that can keep their numbers down are going to be incredibly important. And so we can see actions on the brain, actions on the fat and on muscle cells that can start to make metabolic effects that can be important for protection, including that change in body temperature. If I talk about a change in body temperature, increase in body temperature to stop microbial replication, what does that sound like? Or what might be another word for that? What is it? Fever. That's your fever. So in fact, many of your inflammatory cytokines lead to the effects you think of as being sick. In large quantities, those inflammatory cytokines, your IL-1 beta, your TNF-alpha, your IL-6, can do the things we've talked about, the acute phase protein production, mobilization of lymphocytes from the bone marrow, but they can also enter your circulation and start to have effects like fever, like making you feel sleepy, like making you lethargic and have no energy, like giving you muscle pain, giving you no appetite or nausea. All of these things might make you take it slow so that you actually can conserve energy towards fighting the infection and so that you're not going to get further infection as opposed to going out. And so in fact, oftentimes the things you think of as feeling sick is not something that a microbe did to you. It's something that your body is doing in order to combat those microbes and to help you with an immune response. And so oftentimes when you feel sick, it is actually your body, not the immune response. Yep, Alina.
So is like the higher temperature of a fever, like, or lower, related to how many like bacteria there is, or?
The temperature of your fever is more related to the amount of cytokine that's produced. That could, as you imagine, be related to the bacteria, but it can be related to some other factors going on in the immune response as well. So if you had a fever and you really wanted to, come to class today, what would you do? What might you do before class? You might take ibuprofen or aspirin. Ibuprofen and aspirin have a very specific mechanism of activity. They act to shut off the enzyme COX-2 and stop the production of the inflammatory lipids from these cells. Hi. So that drug that you take in order to feel better is actively working to block this response. I also just mention here because people sometimes get confused. Yes, all cytokines are not pro-inflammatory. There's all sorts of other kinds too, chemokines, etc. All right. So at this point, we've seen tissue damage. We've seen our resident sentinel cell sensing microbes. We've seen release of chemoattractants, those chemokines. We've seen release of vasoactive factors, things that change the vasculature. You can see that in your textbook, it gets leaky. Like here it's not leaky, and here it is. But I liked the side-by-side view a little bit better. Here it does get bigger, but the other view's a little easier. Our capillaries are permeable. We get fluid in cells, contains complement antimicrobial proteins and also cells. But the final, and in fact, frequently these are neutrophils, as you can see in number 3. But we still need to actually do something to the microbe. And so now we need to, in fact, kill that microorganism. We can get rid of that microorganism with our neutrophil. And our neutrophil is going to do phagocytosis. So, because I think every immunologist knows this by heart, I feel it is important to acculturate you so that you can see it as well. So in this video, you are going to see a neutrophil. It's in culture with some red blood cells, which are these smaller gray cells labeled there, as well as some bacteria. Specifically, this is Staph aureus. And once they let the video start going, what you can notice is that those neutrophils are able to sense the bacteria and eventually perform phagocytosis to envelop that bacteria and destroy it. A couple things I want you to notice. First of all, no, the bacteria isn't running away. It's Brownian motion. But do notice that as our cell moves, oh, maybe I should hit play. Notice our neutrophil is going to undergo a lot of sort of shape changes throughout this process. There is a lot of cytoskeletal rearrangement that's required to actually make phagocytosis work. There's also a lot of signal transduction actually happening here. And the point I want to point out is right here, it actually goes right by one bacteria and passes it to keep following the one it has been following. Because of all of the cytoskeletal rearrangement that's happening, our neutrophil really cannot pivot on a dime to get that closer microbe. It's got to continue moving in the way that it has been moving with those cytoskeletal rearrangements. You can find this video easily on YouTube. Immunologists clearly have too much time on their hands because you can find it set to just about any music you want, like the theme of Cops and the theme of Benny Hill and anything else. And so this is the main way that neutrophils are going to be able to destroy microbes. Macrophages also do phagocytosis. You saw phagocytosis earlier with our macrophage. So the main similarity between our neutrophil and our macrophage is their ability to do phagocytosis. However, what happens in addition to the phagocytosis is going to be the big difference between our neutrophil and our macrophage. So first, let's talk about how exactly that microbe that we saw live unhappily ever after in the video get destroyed. You can see that there's some type of binding between the microbe and our phagocytic cell. and we're going to have rearrangement of the membrane along with some actin to actually envelop that microbe. We're going to make a new vesicle as a result of that phagocytosis process. That vesicle is going to be called the phagosome because it got made as a result of phagocytosis. The phagosome will have a somewhat acidic pH, and it will in fact sort of mature throughout the cell, getting more and more acidic, and eventually fusing with the lysosome. The acidic pH, as well as lysosomal enzymes, will then destroy that microbe. And we're eventually going to get to digestion products that could be released or that can be used as more metabolic products for our cell. You can see another view of this here. One thing that I like about this view is that you can see that our phagosome not only fuses with the lysosome that is full of toxic products, but also will fuse with the granules, which are parts of toxic products. Remember the granules that make neutrophils granulocytes that we talked about last week? Those granules have an important job. They're not just there to help you under the microscope or to help us on the flow cytometer, which they'll do in the future. They also, in fact, contain the toxic products that we deliver to the phagosome to kill that microorganism, so that we can eventually bring our microbe into component parts. One of the other ways that we are destroying these microbes is through a process known as respiratory burst. Respiratory burst officially means the increased consumption of oxygen by these neutrophils. So they are going to start consuming a lot of oxygen. But, consuming a lot of oxygen isn't actually how you kill microbes. You can't make yourself unsick by going or whatever like that. It's not going to work, sorry. The point is what happens as a result of this oxygen, what the cell does with this oxygen. The cell makes some enzymes that allow it to convert this oxygen into other products. One of them is known as the phagosome NADPH oxidase, and it allows oxygen to be converted into the superoxide anion. Additional enzymes can allow for production of hydrogen peroxide, potentially things like the hydroxyl radical, and even things like hypochlorous acid. And so all of these things are also happening in that phagosome. The neutrophil or any phagocyte will have the enzymes that allow them to make these products. We also will see production of an enzyme called INOS, or inducible nitric oxide synthetase, which you can see listed here, synthase, I like to say synthetase. This will allow us to start with the amino acid arginine and go to things like nitric oxide, peroxynitrite, or nitrogen dioxide. All of these compounds are toxic to bacteria. Peroxide is something that you would use to get rid of bacteria. Yeah, if you had it throughout, if you drank it, it would be toxic to you too. But you're not going to have it systemically. You're having it in a small organelle inside of your cell in the phagosome to kill microbes. Hypochlorous acid. Anybody know what hypochlorous acid is? What is that more commonly known as? Yes, what is it, Jordan? Bleach, Clorox. So basically, in the phagosome, we're making all of these compounds that have a lot of antimicrobial activity and are going to kill our microbe. For those of you who've taken microbiology with me, it's the reactive oxygen species and the reactive nitrogen species that we talk about in micro that can be toxic to microorganisms and damage their DNA. It's the same exact reactive oxygen species. The phagosome also contains all of the antimicrobial peptides that we talked about last time. So many of those same antimicrobial peptides we saw Friday that can be secreted from cells can also be contained in the phagosome in order to kill microorganisms that have been phagocytosed. At this point, all of this is really similar to what we saw or what we can see with the macrophage. The part that happens in addition to phagocytosis is what's different. With our macrophage, you saw production of inflammatory cytokines. You saw that PRR signaling, which we're going to spend more time on Wednesday, and you saw inflammatory cytokine production. That's not on the table with neutrophils. Neutrophils are not going to make inflammatory cytokines, or at least not as many, not as predominant inflammatory cytokines. They're not going to really kind of start the additional immune response. Sadly, some immunologists kind of think of neutrophils as the dumb ones. But neutrophils do have a couple other things that they are going to do. One of the things that does happen after a neutrophil does phagocytosis is that the neutrophil is going to die. So this is kind of a simplistic version, is the neutrophil dies. So in fact, that neutrophil can die. It can become phagocytosed by some other cell and provide nutrients to that other cell. So our macrophages can, in fact, clear up those dead neutrophils. You know about dead neutrophils that are made after infection. You are all familiar with them. You have likely all seen them. Dead neutrophils that are in a group after infection are known as pus. If you ever put together a lecture on this material, please do not do a Google image search for pus to show for your lecture, because you will get things you did not want to see. So instead, I just wrote the word. So one thing that happens with our neutrophil is that instead of going through additional processes, making lots of cytokines, it's probably going to die and make pus. But what we've learned is that the neutrophil can actually do something super cool when it dies. Super cool. Neutrophils are able to make structures that are known as extracellular traps, or neutrophil extracellular traps, sometimes referred to as neutrophil nets, although that's redundant because the N in net is for neutrophil. So it's neutrophil extracellular trap. So we can see this production of neutrophil nets. The process of having the neutrophil die while making a net is called NETosis. One thing that you will realize this semester is that with Dr. Dunaway, you guys thought about cell death as being apoptosis, sort of like a programmed signaling kind of cell death, or necrosis, sort of like poof. And what you will, with no programming and no signaling, what you are going to see this semester is that there are actually many types of signal transduction induced cell death. There are many programmed types of cell death. Apoptosis is one of them. NETosis technically is another programmed cell death. The cells are dying, but they are doing something specific during that process. And so what actually happens is our neutrophil will start to break down membranes and then modify histones and chromatin. And as that neutrophil dies, it will expel its chromatin to make a net to catch extracellular pathogens. And so you can actually find this extracellular chromatin material that is trapping pathogens. And some of you are making faces like you don't believe me. This is what it looks like. These are E. coli trapped in chromatin from neutrophils. Trapped in those nets. Here is a neutrophil actually expelling its contents to make a net. So while neutrophils don't have the exciting PRR inflammatory cytokine part of their life, which I, as a person who studies PRRs and inflammatory cytokines, I'm a little sad about, they do get to do something pretty cool. Your neutrophil, you can imagine as being a little bit like Spider-Man. It's just a different kind of interesting function, and they are much more short-lived than are our macrophages. So when you think about inflammation, At this point, you're probably thinking, inflammation's amazing. I love inflammation. I want to have inflammation all the time. Because you can see the positive effects of inflammation. Of course, there's a reason why there are ideas out there about inflammation as a negative. Inflammation in precise locations of your body can be involved in homeostasis and repair and getting rid of infections. Inflammation for long periods of time or more body-wide can start to lead to some real problems. And so if things are going well, we're going to see host defense against infection. But if this goes on too long, if this is unregulated, if something goes awry in this response, suddenly this can become an autoimmune disease, or this can become some sort of autoinflammatory disease. We are learning more and more about different autoinflammatory diseases where you just have too much inflammation going on. They don't totally fall under the autoimmunity category. We might see things like fibrosis with too much tissue repair. We also might see some big shifts in your homeostatic set points, which can also potentially cause some real problems. So just to give you a little bit of an idea, some of this is going to happen as a result of genetics. Some of this is going to happen as a result of some other conditions. So to give you some examples, remember our person who I showed you earlier, who had that injury on the hand and ended up with a red hand and had inflammation in their hand, and then they lived happily ever after because they cleared up the inflammation? Now let's imagine that person did not get an infection in their hand. Let's imagine that person got an infection of their bloodstream instead of one location. If that person has an infection of their bloodstream, they aren't going to have inflammation in one location. They are going to start to be producing inflammatory responses body-wide. They're going to have those bacteria or microbes turning on microbe-associated molecular patterns, or turning on PRRs, turning on signaling, turning on inflammatory cytokines, head to toe, throughout many blood vessels. They're going to start to see changes in blood vessel diameter throughout their body. They're going to start to see changes in blood vessel permeability throughout their body. That's going to lead to some pretty severe swelling, systemic edema. Systemic swelling, edema is a fancy word for swelling. And at some point, as they start to have this swelling, as their blood vessels start to leak throughout their body, they're not going to have enough blood volume in their vessels anymore. So we're going to actually see problems of increased blood volume. We're going to start to see things like low levels of proteins, low levels of neutrophils, and eventually things like collapse of the blood vessels because of this lack of liquid. We can also imagine potentially some coagulation issues. This patient is now going to have severe problems systemically that could potentially lead to muscle issues, that could potentially lead to wasting. And this phenomenon is known as septic shock, where we're seeing this inflammatory response body-wide. And this frequently can lead to death. In fact, this is a very common reason why infection patients die in the hospital, is septic shock, otherwise known as sepsis. This is one of the many reasons why understanding how you turn on those cytokines, or how you turn off those cytokines, or how you regulate those cytokines, is a really important question medically. Because if we could do things like turn off signaling to make those cytokines, we could save a lot of patients in the hospital with sepsis. So this is a huge issue. we can also imagine some of those other effects on other organs becoming a problem. If you were to have inflammatory cytokines, this slide shows TNF as our example, but it could be any of them. TNF does seem to have a lot of particular problems associated with it. TNF started acting on muscles all the time, or very frequently, instead of just once in a short burst when you have an infection. You might get changes to heart output, because your heart's a muscle. You might get overall changes to your vasculature. You might get overall changes to the metabolism of your muscle. And in the end, this could start to lead to things like cardiovascular disease. This could start to lead to things like insulin resistance and types of diabetes. And so we now know that chronic production of these cytokines, so chronic production of IL-6, TNF-alpha, IL-1 beta, other inflammatory mediators. There are a couple other cytokines listed here that I don't want to deal with, but it's the big three that we've been talking about, and things like the inflammatory mediators made by COX-2, all that stuff. If those things are produced chronically, all the time, instead of acutely, in a short burst in one part of your body, then we can start to see a number of different processes. We can start to see cell death and scarring. We can see problems with blood vessel growth. We can see overproliferation of cells. We might see impaired insulin signaling or obesity. These can lead to type 2 diabetes. We can also see some of these changes leading to things like heart disease, atherosclerosis, organ failure, cancer, Alzheimer's, autoimmune disease, autoinflammatory disease, inflammatory bowel disease, allergy, asthma, transplant rejection, etc. Basically, if you can think of a sort of medical problem, I can probably link it to inflammation and chronic inflammation. I will tell you right now, I got 9 of the top 10 causes of death in the world. Someday I'm going to figure out how to take credit for traffic accidents too. Other than that, 9 of the top 10 causes of death in the world are all related to inflammation. And so understanding this signaling that we're going to understand on Wednesday is a really important thing that we might want to do to be able to make drugs to block all of these different conditions or to help all of these different conditions. You might ask, well, okay, why is there chronic inflammation? Why is this inflammation happening? That's something that we're still a little bit unsure about. This may have to do with infections that aren't going away. This may have to do with problems in how you respond to the microbes in your body. So you have microbes in your GI tract right now that are helping you out. Perhaps you might have a genetic issue so that you respond to them incorrectly and have too much inflammation. There also might be some other causes. There is some data showing that certain types of lipids, for example, can lead to inflammation, and so obesity or a high-fat diet actually can be shown to lead to inflammatory disease. We used to do modeling of that in my postdoc lab, where we would actually feed mice cookies. Really, there are high-fat diet biscuits for mice, and they look and smell like cookies. Or we can see other types of responses that you may have leading to this chronic inflammatory state. The 2011 Nobel Prize in Medicine was awarded for our discovery, of PRRs because of all of the incredible promise that comes medically from understanding this signaling. We'll talk about that a little bit more later. There's quite a bit of controversy in the field about that Nobel Prize and who it was given to. One of the individuals in question followed me on Twitter yesterday. And some of this is also genetic. So for example, there is a mutation in certain types of diseases that leads to autoinflammatory diseases. So there are some autoinflammatory diseases where you actually undergo inflammation when you are in the presence of hot water. So like if you take a shower, you get a fever. or other types of autoinflammatory diseases that can happen. Some of them are referred to as hereditary periodic fever, where you just keep randomly getting fevers with no cause. In all cases, those are mutations in PRRs that our patients have. And so if we understand this process, this is why I do research on this, because I think it's really cool, we can actually start to understand a lot of different disease states. Now again, you might look at this and you might say, all right, awesome, this seems like a great response. Why do I get sick? How come this is not enough? How come we need some more immunology? Why aren't 4 days of immunology enough? And sadly, part of this is because pathogens have evolved ways to evade all of these responses. So some pathogens have mutations that allow them to avoid detection by PRRs. The sort of classic definition of a PRR is that can be really hard. You know, if we have a receptor that binds to LPS, the microbe would have to change the structure of LPS. If we have a receptor that binds to DNA, the microbe would have to change the structure of DNA. So the sort of general canonical definition is that can't happen, but we do know of a few examples where microbes have evolved in a way that they can avoid PRRs. So you can see, for example, some types of flagellin cannot be recognized by TLR5. There are some microbes that make proteins that can actually interfere with PRR signaling. That will make a little more sense when we see the signaling next time. So we can see a lot of individual blocks with signaling. We can even see some microbes that make soluble cytokine receptors. So basically proteins that look like a cytokine receptor and soak up all the cytokine. So the cytokine can't go anywhere and do anything. So in fact, that's a pretty common thing we see in the immune system, is microbes making cytokine receptors to steal the cytokines. And then we can see some other ways that microbes are able to prevent being killed here. I'm just going to show you, the last thing says, prevent killing or replication. This is that same thing shown in picture form. So there are some microorganisms. The most famous example here is Listeria monocytogenes. Listeria monocytogenes does not want to be in a phagosome to get destroyed by phagocytosis, by the low pH, by the antimicrobial peptides, by the reactive oxygen species, all that business. Listeria monocytogenes makes a protein called Listeria lysine that pokes a hole in the phagosome and lets it escape. And so some microbes just escape out of the phagosome, thus making this phagocytosis process not useful. In fact, it kind of worked to the bacteria's advantage because it let the bacteria come inside a nice, safe, nutrient-free home. Some microbes also can modify the phagosome so that it either does not fuse with the lysosome or so that it doesn't acidify or doesn't have toxic products. And again, these microbes are now able to survive in this organelle as a nice, safe, happy home full of nutrients, no extracellular things coming to get them, etc. These are common strategies used by Salmonella and Mycobacterium tuberculosis. And so these microorganisms basically modify the cell environment. And so phagocytosis isn't going to do anything in terms of helping us get rid of them. Clearly, we're going to need some additional types of immune responses to deal with these organisms. The one final thing I want to just mention is that there are also, of course, very many clinical consequences of mutations in these pathways. Sorry, Nadia?
On the previous slide,
Yeah.
if they're in the phagosome,
Mm-hmm.
wasn't like the pH the main reason?
So the pH is one of the reasons. Some of the enzymes are pH activated, so sometimes it's pH, sometimes it's enzymes, whatever. They actually are interacting with the phagosome so that it does not become acidified. The acidification happens because there's a proton pump, and so it might disable the proton pump, for example, something that actually stops the acidification. Great question. There are lots of clinical consequences of mutations in these pathways. One really famous one is called chronic granulomatous disease, or CGD. Their patients don't have the oxidases, so they can phagocytose lots of microbes, but they can't make the reactive oxygen species to kill them. And so you can actually look at their neutrophils under a microscope and see them full of microbes. But we can see issues in making many of these different enzymes, in trafficking of many of the enzymes involved in phagocytosis and respiratory burst, all of which will lead to patients having immunodeficiency and generally some kind of frequent bacterial infection. Again, sometimes it's a specific bacterial species. Some of these are a little more broad than the ones I told you about with complement deficiencies last time. So our plan for Wednesday is to talk through 2 additional pieces of this. First, we want to go through the exact signaling between the pattern recognition receptor and the cytokine in more detail. Now you know why it's important and why we care about that cytokine, so we can see the details. There is also sort of a classic response that happens with viruses. This is sort of the classic bacterial response, and I'm also going to talk through the classic antiviral response. So I will see you guys on Wednesday.